Gram staining. General information. Gram staining, also known as Gram's method, is a fundamental technique used to differentiate bacterial species into two broad categories, gram-positive and gram-negative. This method, named after its inventor, Hans Christian Gram, identifies bacteria based on the chemical and physical properties of their cell walls. Specifically, it detects the presence of peptidoglycan, a substance found in a thick layer in gram-positive bacteria. In a Gram stain test, gram-positive bacteria retain the crystal violet dye, giving them a distinct purple color. Conversely, gram-negative bacteria do not retain this dye, and are instead colored red or pink by a counterstain, typically safranine or fuchsin. This difference in staining arises from the structural differences in the cell walls of these bacteria. Gram-positive bacteria have a thick, mesh-like cell wall composed mainly of peptidoglycan, while gram-negative bacteria have a thinner peptidoglycan layer. Staining mechanism. Gram-positive bacteria have a thick mesh-like cell wall made of peptidoglycan, 50 to 90% of cell envelope, and as a result are stained purple by crystal violet, whereas gram-negative bacteria have a thinner layer, 10% of cell envelope, so do not retain the purple stain and are counterstained pink by the safranine, fuchsin. There are 4 basic steps of the Gram stain. Gram stain. Applying a primary stain, crystal violet, to a heat-fixed smear of a bacterial culture. Heat fixing kills some bacteria, but is mostly used to affix the bacteria to the slide, so that they don't rinse out during the staining procedure. The addition of iodide, which binds to crystal violet and traps it in the cell. Rapid decolorization with alcohol, and counterstaining with carbylfuchsin. After decolorization, the gram-positive cell remains purple and the gram-negative cell loses its purple color. Counterstain, which is usually positively charged basic fuchsin, is applied last to give decolorized gram-negative bacteria pink or red color. Gram variable. Some bacteria, after staining with a Gram stain, yield a Gram variable pattern. A mix of pink and purple cells are seen. The genera Actinomyces, Arthobacter, Corynebacterium, Mycobacterium, and Propionibacterium have cell walls particularly sensitive to breakage during cell division, resulting in gram-negative staining of these gram-positive cells. In cultures of Bacillus and Clostridium, a decrease in peptidoglycan thickness during growth coincides with an increase in the number of cells that stain gram-negative. In addition, in all bacteria stained using the Gram stain, the age of the culture may influence the results of the stain. History of Gram staining. Hans Christian Gram, a Danish scientist, developed this technique in 1884 while working in Berlin. Interestingly, his initial goal was not to differentiate bacteria but to enhance their visibility in lung tissue sections. His discovery revolutionized microbiology, providing a simple yet powerful tool for bacterial classification and identification. Poem for Gram staining. Gram's method, a name that echoes through time, unlocking secrets in every clime. Peptidoglycan, a wall so strong, in positive cells where it belongs. Crystal violet, a dye so bright, staining the cells with all its might. But negative ones, a different tale. Their walls are thin, their colors pale. Safranine or fuchsin, a counterstains grace, revealing their forms in a reddish embrace. Through the microscope, a world unfolds, of tiny creatures, in countless folds. Gram's legacy, a gift so grand, to see the unseen, throughout the land.